And thanks to Andrea for slides today. So first of all, can we predict structures from first principles? Historically, the moment of showing that proteins fold just in solution, all you need is the chain in the solution, as Christian Anfinsen got a Nobel Prize for that in '72. That means that in principle, if we have the amino acid chain, the sequence, we should be able to predict protein structures. And that brings us to this issue of, can we predict folding? And in fact, you know, there are solutions. So the problem is a very interesting one, but a problem that has been solved. It has been solved. There was a publication in the 60s in the Washington Post. The problem folding problem has been solved. There was a publication in the New York Times in the 70s. The problem has been solved. And there was another one in Washington Post. And the reason why it's those places, because the groups who had solved it came from that local area historically. And obviously, if you solve the problem 3 times, there's something wrong with the solution, right? Now, how would you assess prediction performance? How would you do it? So, mind you, the idea that we typically do in machine learning, where somebody gives you the data set, you take this data set, you split it into 2, one you hide under the table, you pretend that you don't know the data, you develop your model on that, and once you have the model, you take that out and see how well it performs. That's exactly what people did. That led to the mistake. That led, all of these publications here are not people who cheated. So it's not people who deliberately tried to fake it. Those are people, scientists, who really believed. So the journalists completely understood the scientist, and the scientists really completely believed they had solved the problem. They had made a mistake. And that is because once you have those data under here, it's very difficult that you don't peek. It's not that you're actually looking at the data, you're just looking whether it's still there, right? Or something like that. So, and somehow there's a magical way in which this happens to all of us, in which we believe we have never looked at it, but somehow the information got onto my computer, right? And this is the problem. So how could we avoid that? What could we do so that that wouldn't happen?
You do like to do the CASP.
Okay. So that's the, if you know about CASP, then that is the CASP. Here's the CASP idea. CASP, critical assessment of structure prediction. And so the way it is done is the organizers have a period of time, say between April and May, in which they approach experimentalists, ask those experimentalists, is there a protein structure that you're gonna solve soon? You don't have the structure of the protein yet, but over the course of the next few months, you will have the 3D coordinates. That means that when, in fact, the prediction season, June to August, is opening for that particular sequence, nobody in the world, except maybe the people in the group of the experimentalists, knows the structure. But it's not public. It's not in a manuscript. It's not published anywhere, right? So the structure does not exist. In that sense, it is something that is for which a prediction really is a blind prediction, because nobody at the moment of prediction knows. The deadline here is set such that the experimentalist says we were going to do the structure in July, then the deadline is in July. And sometimes the structures, most often it takes longer, but sometimes they actually come before the deadline, and then the case is suddenly closed. For some of the prediction cases, the predictions are no longer allowed, although the initial deadline was put at a different level. Then there is a period here between September and November when the so-called assessors, I call it divine, so they meet and discuss how well the groups did. And then there is the meeting in December in which, in fact, the results are discussed. When we look over time, you see that many, so this is 94 when it started, CASP 1, predictions in red, number of groups, both clearly increasing, and I'm just looking at the first 6 CASP meetings here. Over the first 6 CASP meetings, and that still is in principle true, the thing that really works is comparative modeling, homology modeling, and I talk about what that is a little bit more today. This implies, and I'm sorry for the setting here, there's no generic prediction of 3D structure from sequence yet. I'll get to that again in a minute. And there are important improvements in many fields of structural prediction. One thing that happened immediately after the first CASP is that everybody used the method of everybody else. So that led to the situation of meta servers. So servers that collected all the results from servers, and meta servers that collected the result of our meta servers. And the winner always was the latest meta server. This was clearly one thing that CASP had to fight with in the beginning. That problem apparently is solved now. What I'm going to show you here, slide from the prediction center, from the CASP 12 meeting, where target difficulty, more difficult to predict is here, more easy to predict is here. This Y scale gives you the accuracy, so the degree to which you essentially get the right three-dimensional structure. This is the field of CASP 1. For very difficult targets, they modeled somehow correctly 20%. For very easy targets, they model more than 90%, okay? The first 10 years bring you to the green line here. The next 10 years bring not that much increase, but then you see a jump over these 2 years. And a lot of these increases have to do with the increase of the wealth of the information in the database. This slide here is done at CASP 12. CASP 13 was December 18, and in CASP 13 there was apparently a breakthrough. So I've not been in the meeting. This is something that I learned from emails and then also the science publication here. Google's DeepMind, the method called AlphaFold, aces protein folding, so actually solves the problem. And clearly, that method was a splash for CASP in many ways. So in one way it was a splash because a method that was not available for anybody. And that nobody can easily reproduce because the method is not described, and even if the method would be described, very few people would have the muscle in terms of CPU to reproduce that method. So that we know as much about that method. So some aspects of this method are clear. The other thing is, this was the first time that somebody got to this meeting without any deeper prior expertise in the field. The group Google, essentially, not experts in protein structure prediction. They had used many of the elements from protein structure prediction, but they had created something very new, very different, that clearly was obviously the best method at the CASP. Again, the official results are not out, so there's no place on the website where you can see Google won. This is press, although it's science here. This is clearly the way it was seemingly presented at the meeting, but the official solutions or the official papers are still not out. But here's from Andre, from the prediction center here, a plot that shows essentially the degree to which something is correctly predicted. The line is such that so every point here is one particular CASP target. One thing that it shows you is that at a typical CASP meeting, the number of proteins that you get this way is not in the thousands. Okay? You get a fairly countable number of points here. And if the gray point, each one of those is one protein. If the gray point is above the red line, AlphaFold was the best prediction on that target. If the gray point is below the red line, there was another method that was better. So you see two things. You see that most of these points are above the red line. So alpha really did better than everybody else, seemingly better than everybody else. And there are examples where other methods, although AlphaFold works very well or works best, other methods have their strength. Again, this is a splash, and that brings us to this issue that I already discussed. On AlphaFold obviously does it through machine learning, deep learning. There's the computer, the set of computers from the Shaw Institute, Anton, that is hardware specialized to do molecular dynamics simulation on a large scale. And these, in fact, can now fold proteins up to 40, 50, 60 residues. 60 is still an issue, and we still don't quite know, but for very short things. So this means that MD does not get to the structures, the proteins that we typically have in the database, or typically have in human. But they get already to some exceptional proteins, and that's an immense breakthrough. Okay? And that breakthrough was only possible through, yes?
How fast do they do that? Can they do like one protein a day or one a month? Or?
I actually do not know the answer to your question. So the last publication, I'm also losing time on this, Paul. The last publication they had in which they folded vimentin, that's sort of 30 residues long. In that one, I believe, my understanding was they essentially had done half a year on that. But that is almost 10 years ago now. I'm not entirely sure how long that ago is. And I'm not sure until 3 or whatever the latest model is, how much they can do. They focus more, so I have lost a little bit of touch with these people. I believe they worked more, and that let me get one more step, and again, this is also going to be something that may come in the lecture later, depending on how it goes. But so while we cannot use molecular dynamics to really fold proteins completely, once you have an experimental structure, there's still this issue, where are the dynamic places? How is the function of that? And for that, dynamic programming absolutely works. And that includes this issue, I have the structure, and now I want to have a docking molecule, a drug. So how do I best fit this, and can I simulate that? And that's where Anton mostly goes, because that's where the pharmaceutical industry is. So they are not so much into doing the longest protein possible, they are much more into sort of seeing how that can be used for understanding molecular biology in that sense. Okay, we can in principle do it for short things, but we cannot do it on the larger scale. On the larger scale, one solution appears to be deep learning, and that is news. And this is really news. So this is something that is 6 months old or some. The world was very surprised to see that. So again, there are several people such as David Jones from UCL in London, heavily involved in that. He's somebody in the community. Martin Steinegger is involved for presenting the sequence alignment. So there's a bunch of experts who contributed to that, but it's a relatively recent event. It's something that was sort of not clear summer last year. It is also true that now people sort of, at least some people are known to sort of try to put the whole framework of protein structure prediction methods into the deep learning framework. But this is the moment in which deep learning is sort of really entering this field. Getting back to CASP, there are some issues with CASP. And the one is sort of a perceptual issue that mostly they control now. I just want to sort of flash it to give you an idea of a typical mistake that people do, and not only in CASP, but also in CASP. Say you have 100 proteins that you recruited for the CASP event. Say you have method A that uses 53 of those and reaches some performance X. And you have method B that uses 35 and reaches performance X plus Y, where Y is a positive number. So X plus Y is a larger number than X. Now the question is B better than A? So you cannot say, right? This one is clear. There are a couple of issues. So bring up some questions. So the first question, of course, is the one, is 100 enough? Is it enough that Y is a positive number? So say there is this story, and I can just bring it up to today, of a standard error. What is the standard error measuring? I assume that you all have heard of a standard error. What does it measure?
By how much percent or by how much you expect to be wrong on the typical, kind of like defining a range of results?
Yes. That is somehow measuring the degree to which a number that you estimate is right or not, and typically this bases on an assumption. It bases on the assumption that you have some distribution. So this distribution could be an experimental value, so you measure this value, so let's call the value x here, and when you sort of have a way to check with a more refined measurement tape, so to speak, you see that the more refined values are between x minus sigma and x plus sigma or something like that in 66% of the cases. True standard deviation, 66% of the cases. And then the same is true for performance with the estimate of a prediction performance. So standard error ultimately is something that you derive from a distribution of guesses, so to speak. You never guess correctly a prediction value or an experimental value. There's always some distribution around how right that is. And the standard error measures how right or wrong that is. And that ultimately, the standard error of these estimates for performance for differences of two methods gets smaller the more proteins you have in your data set. So 100 is a relatively small number, so maybe y has to be larger than something because y is just the standard error, right? So y has to be clearly larger than the standard error. That much is clear. But there is something else. There's this issue here. Method A has used 35 cases, and method B, or the other way around, 53, and method B has used 35. Is that okay? Ideally, somebody who has not been in the lectures last semester. Because in every one of my lectures, this is an issue. Yes?
I was just stretching.
Oh. I diagnose success.
I mean, that it uses less cases can either mean it isn't capable of taking in more information, and that method A might be better with more proteins, or if it uses less cases, that could mean that it takes just generally more information from less cases because it's more intelligent, or I don't know.
Yes, that's true.
More interesting algorithm.
Yes, it's all true. It's all true.
It could be good or it could be bad.
So my question still, is it okay to compare them on this?
How can you compare them at all?
Well, that's what we do, right? So in the algorithm that I proposed to you, I said this has performance x and this is x plus y.
Yeah. Right.
Yeah?
We still don't know nothing about the cases. We don't know if they're the same cases or how they're
That's a very
variants in the cases.
It's a very, very important issue. So I said that the original set was 100, right? So in principle, you could add up these numbers. You could add up these numbers such that there's zero overlap between these two numbers. 53 plus 35 is less than 100, right? So I'm not saying that at all, and that brings us. In fact, in this particular case, we actually, if you think about it, we compare apples and oranges. In the most extreme case, we would completely have different objects that we compare, completely different parts, slices of the 100, right? Which could be, in a sense, what Paul says, could be one works on the simple cases and one on the difficult ones. In that sense, as Paul says, there's something interesting at least in this comparison, right? But this we have to find out. Now, this leads, something like that, leads to a situation where we get a wrong ranking, yes?
But isn't the onus on the creator of the program or of the algorithm to use all information?
Well, that depends. You could have a method that is specialized on something, right? You could say, I am very good at the high accuracy homology modeling, where I really simulate the change of side chains. That's what I'm good at. And somebody else says, well, I don't care about that one. What I care about is finding something that is sort of roughly the same. And my specialization is in finding something that you will never find with your program.
But then it's a poorly defined problem.
So then you would call it two different classes, and that we should. Well, but that gets to the idea, how do you group? Yes, so the way CASP has been deciding on these groups has changed over time. Because then the groups began to overlap too much, they were merged, then they were sort of changed, they were taken apart by essentially the value. You saw this axis difficulty. So in the olden days they had different classes and they had different names, and today there's just a scale. And in some sense, this is sort of the correct solution, but as you say, then this is a mistake because you no longer have classes. So it's a complicated story, right? It's a problem that, so this is not an issue that you can solve. It's not a responsibility, the onus is to the organizers. This is not solvable. And ultimately, the response here to have a gliding scale, I believe, is ultimately exactly the right one. You're not arguing method A is specialized on that, method B on that. Let's just see whoever contributes to that level and how well do they do in that level. But it brings up a new issue. And the new issue is that we have a very limited number of proteins. We have 100. And if we are not talking about 2 methods only, but if we talked about 10 or 15 different methods, and in the following one, I believe I have 7 or 8 methods, and I'm going to show you results for one of the earlier casts when secondary structure was still one of the disciplines. So then there were disciplines, categories. And in these, the methods, so it's a sort of simulated result, but it was true that methods did predict for different proteins. So what we had, so one way in which we can cut this story of 53 and 35 would be to say at least the number has to be the same. We still have your issue. We still don't know whether there's any overlap, but at least the number has to be the same. We're not allowing that one predicts 1 and one 53. Okay? At least they have to predict a similar number. And then if we, the lowest or the highest number that all of them had in common was 29. Again, forget about the overlap, just 29. The method of the 8 that I show you with the fewest predictions had a 29. So the lowest number, the highest number we could take was 29. If we actually looked under the hood and asked how many actually are overlapping, then those were 11. So now we have the choice. We can assess methods on the 11 that are identical, or we can assess them on the 29 that it knows they have predicted that will not completely overlap. Only 11 in these 29 will be identical. Okay? Which one is going to be better? So the criterion for better is, now in this particular case, underneath I had a method at the day, we had an automatic server that collected over years the results from these 8 methods. So these 8 methods had predicted at CASP, but we had also results from 3 or 4 years of pseudo-blind experiments where we had always used the latest targets that PDB had released, but they were not available to the servers at the time which we sent it. So we had a glimpse at the sequence before the structure was available. So servers could have taken it from the internet, but on the level of thousands of proteins, nobody did that, okay? So we had an automated procedure that sort of simulated CASP, and this means we had the real ranking. We knew the real ranking after 5,000 proteins, and now my question is, correct or not? So which of these 2 solutions gives the correct solution? And correct is, I have 5,000, I know what is correct, the ranking, right? And I'm asking, if I only had the CASP, then that is, at that particular CASP in which we sort of did this, we had these 2 options. Which one is better? So you compare methods, you rank methods according to 29 proteins that they predict for. So they all predict at least for 29, or 11 that are identical between them.
I still don't quite understand why the other one does, why they don't predict the same protein. They just not get a result.
So we don't know. We do not. Yes, there's a variety of answers. The deadline is close too fast. The protein is too long. That protein is a fly protein. That server doesn't model fly protein. There's a variety of different answers to that question.
Oh.
They started the competition too late. So I'm mixing 2 things here. So what the reality of that CASP really was that the servers that we had at that CASP, the maximum number of the 100 was a 29, and the identical one was 11. So those numbers are real from that CASP. I do not know why. I do not remember, but it really boils down to a variety of different answers. Some of those in the one that you call interesting because methods focus on slightly different aspects, and some of those really server broke down, format issues, this or that. Strange letter. Yeah? So which one is better? Which one would you take? Okay, you have 2 choices, 29 or 11. Who's for 29? Who's for 11? Everybody else leave the room. You have to say something. Wow. Really? 11 is a totally tiny number, no? You're rather... Okay, so you guys are somehow propped in the wrong direction. Many years of CASP, they did 29. So actually many years of CASP, they did the 53 to 35. And then they started using the 29. But going this jump to 11 is an extreme jump. You have to understand that every structure is a value, that every experimentalist the organizers pursue is a lot of email exchange and a lot of coffee you have to pay or whatever. So this is not for free. And now you suddenly throw away 89 of these. You just say, well, okay, your 89 were great, but you know, we only use 11. But ultimately that is in fact the right answer. You all are on the right side. So I sort of, essentially the way to think about this is I'm simulating 30 different consecutive CAS meetings here with 29 proteins, and I simulate the same with 11, and the spread here is less than there. That ultimately is the result. But it is also true that there is an immense spread of the ranks, and that none of them really gets anywhere close to the real image. And that sort of gets to the solution that is now implemented in CASP, and that clearly is in advance. In this solution, what you do is you have your 4 methods in this particular case here, and you essentially do a pairwise comparison. And for every pairwise comparison, I said they have 11 in common, all of them. But every pair may have many more than 11 in common, right? For every pair comparison, you ask, is A significantly better above the standard error, right? Substantially above the standard error than B. If so, then this is blue. It's a much better. Dark blue is 2 standard deviations. Maybe that's 1 standard deviation. That is indistinguishable. Or that is indistinguishable with the gray, or the gray is just grayed out. That's indistinguishable, and that's substantially worse, right? So it's symmetric, I mean the inverse color. And this is essentially the way it's done today. Now, this is still not clear how you get from here to ranking. So one way you get to the ranking is you say the one with the most dark blue. Another way to the ranking, you say, well, as long as I cannot distinguish, for instance, I cannot distinguish A and D. So they have to occupy the same rank. Which means that possibly I could distinguish between, let me see whether there's an example, between D and A, there is a distinction, so this example doesn't give it. But it could be that, let's just make up an example. So you have an accuracy, whatever that means, of method A, which is 72, and there's plus minus 1 as a standard error, and B has, say, 70 plus minus 1, so they can, well, or 71, and C has 69.5 plus minus 1. So between A and C you can clearly distinguish within 1 standard error. Between A and B you cannot, so that's why you have to rank B as number 1. And between B and C you cannot, that's why you have to rank C also as number 1, although A and C are clearly distinguishable. So this is another way of doing it that creates a ranking that has issues. Yeah?
Why isn't this metric symmetric? Like, why can't we distinguish between A and B, but we can distinguish?
Yeah, it ought to be symmetric. I stumbled over the same thing. There's absolutely no reason. There is absolutely no dissymmetry in this matrix. Again, it's really, has nothing to do with the age of the slide. I put this in incorrectly. It's absolutely nonsense. So the only way I can explain it is that over time, the computer suddenly made it white. What can I do? I made a mistake. And I don't understand how I can in 16 squares have one so...
Well, even B and C, if one is red and the other...
No, there's absolutely none. No. No reason. There's no reason. It's a mistake. It's an honest mistake, and I will just forget. Thank you for being awake. I just stumbled over it too. Anyway, idea is clear. So how do we do structures from experiment? I already talked about that. In 2017, the number is higher now. There are 120,000 protein structures. About 90% of those come from X-ray crystallography, 9% from NMR, 1% from EM. X-ray crystallography. You begin by getting the protein to form a crystal. Now, is that easy? Do proteins look like crystals in the cell? Actually, Paul, you're too fast. There are proteins that are in such high concentration in the cell that you can actually almost, no, not almost, you can actually scoop them out and have a crystal. There are few exceptional cases. Most proteins are not like that. But remember that really the density in a cell is of you think about a cell more like a watch. So the density is really so high, it's actually not that strange. But still not easy, okay? So it all begins with a nice crystal. So if you're a crystallographer, you show an image of a crystal and then you find this looks beautiful. Actually, it looks beautiful. And the crystallographers or people who are experts in this field, they look at these crystals, even at these photos, and they can tell you something about how good that is going to be a structure. Just from looking at the crystal. I still haven't sort of quite acquired that ability, not at all actually acquired that ability. So you shoot X-ray, very highly parallel X-ray beams that you only get from synchrotron sources, of which you have very limited number, at the crystals. From the phase of the diffraction pattern, and that ultimately is when you go to the subway, there you see the idea of how you actually translate an image, how you do this dephrasing from Bragg. So you create phases that ultimately give you as experimental data an electron density map. So defining the probability for where the electrons sit. This electron density map you use to fit the atomic model. As a model, because, and I'll get back to that in a few slides from now, there are sometimes some alternative options that you can do. Again, 90% of the structures are like that. The first one was done like that. This is sort of the most straightforward way. The problem, there's a variety of problems, but one is actually to get the crystal. NMR, nuclear magnetic resonance, starts from the protein in solvent. You just take it as it is in its natural environment. So that sounds very, very great. The trouble is you need big magnets. So the synchrotron is also a big machine. But in this particular case, the synchrotron is one that you have, I don't know how many we have in Germany. People do it in Hamburg. So the major one in Germany really is at DESY in Hamburg. So there's one. These machines here, there's a smaller version of this size here actually on campus. So there are magnets on a scale that individual labs have, right? This room here is a fairly large room at City College, the New York Structure Biology Center. There are a few of these large centers, machines just to look at the scale. This is a person up here. These magnets are really gigantic and very, very, very strong. What you then get is alternatively possible models that are compatible with the data that you get, as shown in this particular PDB structures. When you look at PDB, you see that for NMR models, you're not only having one set of coordinates, but n. So n here, 1, 2, 3, 4, 5, 6, 7, 8, and so forth. That's the number of models you will have. And you see that sometimes in some places they differ, and other places they are identical. What's the interpretation of that?
They differ, the protein is more flexible, and where they are identical, it has to be in that structure. That's
exactly the idea. So it could, of course, be that there's a mistake in the measurement. That could also be the reason for the differences. But the interpretation is that where they differ because of functional mobility, that is where we find different models. and where they're always the same, and they need to be that same for the structural, for the function, that's where they're all the same. That means we are not only getting actually something about the structure, we're also getting about the dynamics of the structure. So this additional is knowledge, if you want. That's the NMR story. If you go one level simpler in terms of experimental setup, you look at a cryo-EM. There's an electron microscope. Cryo, because you have to freeze the protein. You don't have to grow a crystal, you just freeze it. Now the problem is you're actually looking at, with the electron microscope at the protein, and what you will only see is the surface. Depending on the resolution, that depends on that particular experimental setup, on that particular protein, on a variety of small things, details. You see something that's sort of 32 angstrom resolution to 4 angstrom resolution. And on this level you see sort of holes. On this level you begin to see really molecules. You still see only the surface, right? You still don't see into, that's Groyal, you still don't see into the Groyal, into the chaperone. But you could now imagine that you sort of combine this outside constraint with inside modeling. And this is one solution from the from Chu, where in fact there is comparative modeling used inside or other modeling. There's a variety of groups that essentially sort of combine EM with modeling. By the way of EM, Joachim Frank is one of the people who have really pushed the development of EM modeling. And there will be a talk from Joachim, I believe, on the 6th of June at 2 o'clock. Joachim Frank in the IAS. Joachim Frank is also last year's Nobel Prize winner. Okay, now back to the resolution story. When you, and I'm sorry for the quality, I'm too lazy I actually cannot make it dark enough. You may see that there is a light blue. Can you see it at all? If you see it at all, you see that here at 3 angstrom, there is sort of a long extended rod into which that is all the electron density in blue. Here, the rod actually is more of a sphere. So while here it's one long rod, here is a sphere. Meaning that the way you orient the ring in the rod leaves you a lot of freedom of movement in that direction, while here, at least you cannot move in that direction, right? You can still move in that direction. And the most extreme one here, for 1 angstrom resolution, you really see the ring. So there is no arbitrary solution anymore. This, in fact, completely nails every position in the atom. Most structures, there are more structures on the level of this one here, increasingly on this level. This still is the exception. But this is the other extreme of resolution. So EM, electron microscopy, is really not on the level. But EM can do large machineries. Again, in terms of 3D details, I showed you different aspects to really understand how it looks. You look at secondary structure. Secondary structure is stabilized by hydrogen bonds, and that ultimately is a statement that was first proposed by Linus Pauling and got him the Nobel Prize in Chemistry in 1954. So essentially you have a hydrogen bond formation between residue I and I plus 4. And I plus 1 and I plus 5 and I plus 2 and I plus 6. So you have essentially to every fourth, and that, bless you, and that creates the helix. The beta strand is every other residue. So this is residue I plus 1, I plus 2. So every other residue points to the other chain and forms a hydrogen bond in the beta strand. Now, again, this is sort of Linus Pauling gets the Nobel Prize for understanding that secondary structure formation is determined by hydrogen bonds in 54. So when was the first structure determined? It was on a slide before, but I hope that you haven't seen it. So you get the right idea of that. In fact, not only do you get the right idea, Kyla, shockingly you get exactly the right number. How did you know? You saw the previous slide. Thank you very much. So one of you is awake. In fact, that is the structure from Kendrew, myoglobin. Hemoglobin is from Max Perutz 2 years later. I have to say, really the whole story started with Max Perutz. He was the one who pushed the idea that we can actually determine the structure of a protein in the 40s already, and that was his sort of postdoc. The structure came out earlier from Kendrew simply because he was lucky enough to work on something that is faster to get a structure for. Can you guess why?
Because they had so much of it.
Yeah, that's an important point. But actually, so they went for the myoglobin and hemoglobin both bind oxygen because in fact they can relatively easily get a lot of it from blood. No, but just look at the images.
Because it's small?
Well, it forms a dimer. So the point is you have to form the dimer, and that makes it actually bigger. The individual molecule is similar, but that was exactly the entire advantage, and that's why he got a structure much faster, and sort of he piggybacked on the machinery that had been developed by Max Perutz. But there's something else that you may notice from these structures in terms of secondary structure. It is only alpha helical. So structure number 3 came out in 67, was all alpha helical too. So you can guess where the story went. So people believed that the idea of a beta strand is nonsense. We will never see a beta strand. For many years, people really believed this is all we will ever see. Today we know there are beta strands. How can we annotate 1D secondary structure from coordinates? How could you do it? So I give you a 3D structure, and I want to get secondary structure out of it. How would you do it? The first principal ideas of the 2 first methods uses geometry, angles, and DSSP uses essentially an energy. In the energy, you simply have an energy function that says if 2 atoms are close enough to each other so that they can do a hydrogen bond. If you have a hydrogen bond, you simply count up I and I plus 4. You call it helix. If you have the pattern of a beta strand between any pair of the protein, you assign that. Again, you find the hydrogen bond and you assign the structure. Then there are sort of other methods that have been proposed. I want to mention STRIDE because it comes from Dimitri Fishman, and that essentially tries to machine learn expert assignments. Predictability, if I want to make that point because it's an interesting idea. So just assign the secondary structure according to what you can predict. I find this, in some sense, you can turn the problem around, but it's an interesting idea. And then there's the continuous secondary structure. So that says that one issue with secondary structure assignment is in this idea that Kyra, right? Kyra. And your name is missing here. No. This has to do with, let's say, hardware issues. Where your name is, there's a big white blob because our printer no longer works. So one issue here is that in Kyra's proposal, you have to say there's a certain distance, and that distance I say there's a hydrogen bond. That's the threshold. If it's a little bit more or a little bit less, chemically, it's a probabilistic hydrogen bond. Sometimes it's there, sometimes it's less, but the bond somehow exists to some probability, right? And this is the idea behind the continuous DSSP assignments. So there are cases where this hydrogen bond will always form, and then there are cases where it's a probabilistic measure, and this is reflected in a residue not being part of a helix, but being part of a helix to 90% of the time or something like that, right? That's the idea behind this one here. The 3 earliest methods, there was a paper published a long time ago from Nathalie Kollock in which she showed that the major 3 initial ones agreed only to 30% of the helix assignment. Whichever that is, today essentially everybody uses DSSP, which is essentially the idea of Kyra. Or Kyra. Comparative modeling or homology modeling. So again, we in principle know a sequence adopts a unique 3-dimensional structure, and that sort of implies that similar sequences, or may imply that similar sequences have similar 3D structures. It is not necessary. Here's an example for the protein oncogene KRAS. slides come from Andreas Schaffer-Hans. And here's the same KRAS with one mutation. So the glycine at position 12 is mutated into a cysteine. Now that change, and you see the superposition of both structures. What you see essentially is the same thing. You don't see two structures, which means they are essentially the same. You see a tiny difference here. And you see that the binding is slightly different. In fact, that variant, G12 to C, causes cancer. Or it's the one that ultimately relates to some people why this is called oncogene. Let's put it like that. But still, it's a relatively subtle change. Now, if we went on in terms of sequence identity, went down on in terms of divergence to 85%, 15% of the residues now change to Rosh, then we see that we begin to have changes. But it's still a clear helix, and still even in many places the helices are identical. Still the binding site is relatively similar. Even some loops are very similar. So there's a lot of similarity. We go further down to the level of fly, 28% sequence identity. So we changed 72. And we still see regions that are largely very similar, still similar binding site, although we changed 72% of all the residues. We go even one further into the bacterial world here. We get to a protein that no longer really has the function, but still looks quite similar. By the way, this is sort of sampling of that family, structural family. Most of the structural family are bacteria. That's what this one shows. But ultimately it shows that you can change residues and you sort of maintain still structure. One way in which this is expressed is this HSP curve, where you show number of residues aligned, percentage sequence identity. Every pair of proteins, if naturally evolved, that is above the line, essentially have similar 3D structure. Again, if naturally involved. You could simulate or you could create sequences that are above the line that are different. But sort of naturally evolved ones above the line typically have a similar structure. And again, there's some sort of unclear region in between, but ultimately that statement is true. And that means that now comparative modeling sort of essentially targets these cases. The way it does it, essentially, oh, by the way, I'm stumbling over trying to get through too fast through this. We say that we can predict structure, or we can use structure to learn something about function. One way in which we can do that is the method conserve here from the Nirbental's lab. Simply colors the conservation. Conserved means dark red, variable means very strong blue, onto the structure. And you somehow see that wherever it's dark red here, you in fact is exactly where the binding site sits on the surface, right? Binding on the surface. So this is one thing that you can see. And this sort of thing you can actually see even for, if I look back to the RAS example, for the rush or for the fly protein. We would still sort of see a similar coloring. While in some cases, and that is an example here, the archaeal structure, when you just look at the feature that is plotted by Darren Murray here, if you only plot sort of the electrostatic feature of the model that uses the same 3D coordinates between the human and the archaea, the electrostatic features look very different. Blue versus red. Same 3D structure. How can that happen? Actually, one in this particular case is the model from the other one. So using the same 3D coordinates, how can that have a different electrostatic surface? How can that happen? If somebody feels cold, I'm going to close it, but I just feel that air is needed. There's some signs in the group that suggest aeration helps. Yes? Any idea? Any suggestion? It's actually quite simple.
Different amino acids?
Yes. All you need to do is put at some point a charge, right? So charge at the same position in 3D structure, put some positive or some negative charges, and one immediately will change the electrostatic potential. With very, very few residues you can achieve that. Structure looks the same, but the functional features are different. So you can use actually a comparative model or something that you sort of is very similar to the other one and still see differences in function. And that's an example where that works. Ultimately then the idea of comparative modeling is you have a set of structures that you know, and you have a sequence for which you do not know the structure, so find the one essentially that looks like yours. And the way to do that is you begin with a sequence, a protein sequence, you find a protein in the PDB of known structure that looks like this, for instance, and you say the sequence similarity is enough so that I believe they have a similar structure. Okay? The reach of comparative modeling, and I'm going to go through this. This is too slow for me. Ultimately, we can model a little bit more than half of all the data. That's just one point. A lot of the things that have similar structures we cannot find, but we can sort of, this is percentage of genome that we can model, which is slightly less than half. But today, this is sort of ballpark, let's say just half. Half of all the proteins we can model. Okay? That's the take-home message. And by the way, there's one other issue here, and sorry for the animation. So this is as much as comparative modeling could do in 1991. And I thought this is as much as it can do 1995 and 1999 without changing the method. So this improvement of the blue here, or the increase of the blue, the coverage of the blue, simply comes from the database growing. Not doing anything, at least that is what I simulated underneath here. Let's get back to the idea. There's something about words here, comparative modeling versus homology modeling. People use different words. It's all the same. Essentially, you use something that people call a target to model a template. Target is the protein for which you know the three-dimensional structure. Sorry, the other way around. Target is the protein that you want to model. A template is the template of the structure that you have. The protein, in this particular case, for instance, you may want to model RASH or RAP, and you have the fly protein structure. So, say you wanted to model this one, and you had these two here. You found out from the sequence alignment that these two are similar. Then you could sort of use either of those two. How can you identify the template? Well, all kinds of the alignment methods that I showed you so far would work for that. So into the twilight zone, we can sort of safely align with good alignment methods that use hidden Markov models, sequence profile-based alignment. Profile, we could push it a bit further. Now, then comes this issue of building the model and how to assess the model. So assessing the model, so building the model, how would you build a model in comparative modeling? So you know the structure for a fly protein, and you don't know the structure for the human protein. How would you build a model for the human protein?
There's a method where they create a contact map, and then they basically...
Let's begin the simplest step. Well, what is the simplest thing you can do?
Take that as one-to-one.
Yeah. Sorry, is that what you want? How do you take it as one-to-one? Where do you know what is corresponding to what?
To an alignment.
Yeah, simple sequence alignment, right? The sequence alignment gives you exactly the overlapping position. So ACELL is, say, the human protein. And then you had a GPQII or something. Oh, this is zero similarity. CE. Now we have at least 60% similar identity here. So you essentially take, if that were a fly here, you take the coordinates of the C, put it to the C. You take the coordinates of the I, put it to the L, and so forth. You just simply copy coordinates. Now that may lead to a situation such as this one, because say the story here continued with a lysine here and an arginine here. So one is substantially larger. So putting the arginine at the same position where you previously had a lysine will lead to some clash, some high overlap, right? What will you do?
Start only with the backbone. There is this isolate, then
is in fact exactly what they do, but it's sort of a cheating trick, right? Because so you have a clash, and essentially if you think about, but this is what they do, so you're 100% right. But if you think about what you're suggesting, okay, let's not look at it. Let's just pretend that there is no clash. Oh yeah, there is no clash whatsoever. So, but again, I'm sort of making fun of you, Kyla. The reality is this is exactly what people do. But my question is, what next? So the moment you open your eyes, what do you do then? So you have your initial model. The initial model is exactly this, and you see a clash. What will you do?
And you just shift your...
Exactly. That is an extreme statement. So shifting the alignment, so if you shifted the whole thing over, you actually create a hole. Well, that's a very radical step. What you do see here is overly dense packing, is a clash of atoms. But shifting the alignment means you make a big hole. That's a little bit too much. And even if you did that, you still have to move things around, right?
Where would you get your new coordinates from, though?
No, I'm saying don't use new coordinates. So what I'm saying is move it a little bit, right? See whether you can push in the direction where there's too much overlap. Maybe there's another residue behind that you will then push that one a little bit, and see that you one by one push a little bit without getting too much out of line. So small movements around to avoid clashes, and see whether you can do that. If there are some clashes you cannot avoid, then you accept those clashes. And then you say, this is my best prediction, because ultimately exactly with the rationale that you continued, where will you get the coordinates from? You don't know the answer to that question. So you simply answer, okay, I have a model that is like that, and I know here it's wrong. I cannot tell you what the right solution is. All I can tell you, this I don't trust. But it's the best thing I can do. Right? And that gets, essentially this is the part of the SS model, refined model, in sort of a more generic way. If the template and the target are very similar, then the major problem is the speed of doing it. If they diverge more, and where exactly you put the numbers is just giving you an idea here. The quality of the model becomes an issue, so simply how close they are, the alignment, what fraction of the protein do you model, those kind of things become an issue. And then at some point the alignment issue becomes a problem, because if the alignment is wrong, you copy the wrong coordinates, and there's no way directly to benefit from that, or to recover from that. And then if you go even further, then the issue really becomes detection. So to see that the fly protein is the one that is in fact the structure of the human becomes the problem down there. A few things about state-of-the-art. Modeler is clearly one of the state-of-the-art. Modeler is a program with a lot of bells and whistles that is optimized for people who know what they do. Again, it comes from André Charlie's lab. A lot of what André today does is what I showed you in this image before, fitting structures into EM images and essentially spanning the entire world from data in the world of experimental data, low resolution, high resolution, into getting structures and predicting function. Modeler, Marc Marty-Renom is one of the main people behind it, goes back to work that André did with Tom Blundell. Is the comparative modeling suite that you can actually download. So it starts with a fold assignment, essentially meaning that I identify the protein that is similar to you, aligned target and template, build a model, evaluate the model, ask whether the model is okay, and then sort of try different models, try different solutions, different alignment, different templates, and wobble around. To get into a few of these states is a constrained satisfaction, gives you an idea how good the model is. Essentially, how well do you satisfy these clashes? How do you get these clashes? And that ultimately gets to some of the issues that implicitly you said about secondary structure. What is a clash of atoms in fact, the only way to find out is you measure in the database, you look at some distributions of a C alpha distance, which is the way you suggested to begin. You just look at C alpha, and then you know already a clash on the C alpha, and you see that there is, in the observed one, there's a variety of cases, and you sort of fit it into that. The next issue is that you begin to sort of model into this constrained satisfaction issue, and that takes some time, and over time things get better. This is what that says. And there's a bunch of underlying dynamics that are done, simulations. So when I say comparative modeling, what I describe is really a suite, and I completely hide that there's a lot of details underneath. As you see, there's a bunch of different optimization steps employed. Now, one typical issue are sidechain issues. So the sidechains are incorrectly aligned. But another extreme issue, and I'm not sure, let's just get for the issue, can I identify the right model? So this is the error, this is the internal score that says this is the correct model. The lower that score, the better the model. In this particular case, you see that the lowest score is the lowest RMSD. Then there are cases such as this one here, where the lowest score is not the lowest RMSD. The lowest RMSD would be this one here. So, in fact, they can find the model that is right, but they don't pick it. They pick this, which is more incorrect than this. So then they have sort of a modeling issue. And they do all kinds of issues to get rid of these kind of mistakes over the years. But what if really you had an example where you have a loop, and that loop is in fact missing, completely missing in the template? What do you do? So your own protein has a loop that the other one doesn't have. How can you model that loop?
Maybe try to find a similar loop from a different protein.
That's the first step, and that's exactly what people do. So you look at all, you know, constraints. You know that one end has to be here and one end here. So you have a constraint on the distance of where the loop is going to close. And then you're going to find all the loops that close at this distance constraint. As simple as that, and you somehow find a good average over those that have a similar length in terms of number of residues, right? Another way in which you can do that is you do a loop modeling, where you exactly do a molecular dynamics simulation of loop. I told you that Anton can do it for not too long proteins. Loops are short. For short loops, people can sort of do that. And that works for loops that are not too long relatively well. The other program, so model has lots of whistles and bells, is downloadable, is very accurate. Another tool available is the Swiss model. It comes from the Schwede lab. It's Thorsten Schwede. Very, very present, very, very well seen. Originally behind it also Nickel Gui and Manuel Peitsch. And so in the initial implementation of Manuel's Swiss model essentially did the following. It did a blast to find the closest homolog in the PDB, took the corresponding alignment, copied the coordinate, end. Manuel's logic for that was, if I do not do anything more than that, I make no additional mistake, and I can actually go home and have a free time, ultimately. Now this was, so the way Manuel presented that as one of the first class meetings, people did not like that at all. So, you know, somehow scientists are very active people. And Manuel suggested that he's not. It's not true, but he succeeded in convincing everybody that this is actually what he does. And the problem for the people was that was the best performer at the cast, because everybody had used all their intelligence to mess it up. Not because they were dumb, right? Because the problem is really so complicated. And in fact, indeed, the entire field had added more errors than Manuel, because Manuel had not done anything. Meanwhile, Swiss model. This is also why Swiss model was the first server, because they could do it very fast. And they ultimately did not so much care about the mistakes. They said, this is simplistic anyway, but maybe it's useful enough for you. And with that philosophy, they went online much faster than the others. Today, as I said, alignment with BLAST, copy coordinates, and this is no longer the case. So today, there is a much more complicated tool set underneath that is in complexity very similar to Modeler and differs in detail, and I don't want to go into it. Here's a bunch of other methods. H.H. Pratt, coming out of the lab from Johannes Söding, who is the strong Max Planck Institute in Göttingen now, André Lupasch from Tübingen. Underneath is HMM, hidden Markov model, alignments that profile alignments that cleverly predict structure, and that is in fact one of the best methods available. 3D jigsaw from Paul Bates and Michael Sternberg is ultimately the idea to use the database of all the proteins, chop it up into fragments, and fit fragments together. It's a 3D jigsaw puzzle. That's still sort of underneath that work. This brings it to fragment-based approaches, of which 3D jigsaw is one. What If from Gert Frendt is a tool that is, if you go into that, if you sort of understand the logic of what if, it's very good to model structures, not to look at structures and see how can you change. So the idea about the word what if is, what if I would change this amino acid? What if I put a drug in there? How would the protein react to that? What would be the stress, the energetic stress? And that is what this program allows you to do. And thanks to Emma Krieger, it also does through the method Yazara, it does very nice images. And that's, by the way, is also available. Now there's another one here from Czeslavas Venclovas in Lithuania, Vilnius, that does very well on getting the right alignment and gets many of those aspects right. I believe I leave it there. ITASR, one of the latest class winners is ITASR from the Chung Lab. And again, this one we're going to go over. But the thing that I want to make is when we look at sequence identity between two protein structures, these curves here, that is RMSD, and these curves are different comparative modeling servers from some point in the past. The important issue is there is a range at which sort of most of them have a fairly high accuracy, and then they make mistakes. Okay, but the point that I want to point out, and this is a little bit better today. So the best servers now are lower, and go lower a little bit longer. Okay, that's the improvement of the field. But the important thing that has not changed is this one, and this one shows what fraction of the protein do I actually cover. This essentially goes back to, I have additional loops, and these additional loops just don't correspond to the template. The target is longer, right? And what you see is that here in this realm, until sort of 40%, 80% of the protein is covered. But then you radically drop, which means that this number here is only on 20% of the residues. So yes, you can do comparative modeling, but you can actually do comparative modeling of fairly short fragments. And this is still true today. So another image that I want to bring that comes from René Magritte, it says, this is not a pipe. What does it mean? So this is clearly not science now, the question I'm asking you. The picture of a pipe, and this is I'm bringing that up because models, comparative models, are models. They are not the truth. So something must be wrong, right? There's also this image, the idea that I always have brought up for the first lectures of images. What is reality? What are images? And that is important to remember. Now, Thorsten Schwede sort of said it differently here. Model is a tool that helps you to interpret reality, that helps you to interpret function. And essentially, what you have to think about, models will be wrong. So look at where is the model going to be wrong. Look at what can I trust and what can I make out of that model. What can I learn to predict function. Coverage, we have 150 million proteins for which we know the sequence. About 150,000 proteins for which we know the structure. These 150,000, we can model about half of the 150 million. about 75 million of the sequences, we can model 3D structure on the level of comparative modeling fairly accurately. In many of these cases, it will be a relatively short part of the protein, so that does not mean that we have 3D structures for 75 million proteins, but we have some region in 75 million proteins for which we can predict structure through comparative modeling. That's an immense power. So what I'm talking about is something that is essentially the answer today for structure prediction in practice. However, for the other 75 million, or for those for which I have 20% covered, the other 80% of those proteins, essentially, I cannot predict 3D structure. AlphaFold goes into predicting 3D structure, but so currently the method is not available for you to submit your sequence. There are some servers out there that for some of these proteins you can predict aspects of structure, and they're getting relatively close. But the only thing that we can do for every single protein at this point, reliably, is to go to a much simpler problem, that problem of predicting 1D structure, secondary structure.